In fact, one of the things we're looking at is working on hail, and I'll talk a little bit about a hail if that's okay. But the main topic is going to be blackleg. And since Murray was talking about resistance stewardship, I will talk about that first, and then we'll have a quick little review of symptoms and so on, and then some questions, if that's all right. So Murray was talking about gene stewardship or resistance management. What that really is, as he's just to recap, is really how do you preserve the genetic resources you have, the resistance that you have. So in clubroot we heard there was, it's called a limited number of genes available, and we've got to preserve that for as long as we can until more resistance can be put into varieties, or more diverse kinds of resistance can be put into varieties. And this is a common theme in a lot of plant diseases. And the plant diseases where this happens, where you have to steward those resistances, are diseases where you have a sexually recombining organism, or another way of putting that, something that can shuffle its genes very, very easily. And strictly speaking, that's not true for clubroot, although it does shuffle its genes in a pseudo-sexual way. And that's also true for blackleg. So, and the way the blackleg fungus does that is it has two spore stages. One of them, and you can come up here afterwards and have a look at stuff in this bag. You can see that these are the little woody bits left over after 2 years sitting in the field of the top of the taproot, the bottom of the stem base. Turns into a little piece of driftwood like you see here. And the spores will form. They're called the structures are called pseudothecia. They look like round spheres, dirt, almost like globs of dirt sitting on the stems. And those things are the product of recombination. And these things are what allows the blackleg fungus to rearrange its genes to, in quotes, learn how to affect different varieties or different genetics that we throw at it when we put a new variety out in the field. And when we say try to extend that canola rotation as far as possible, for blackleg, the objective is to try to get rid of these things, to try to have these things decompose. Because once you get rid of the stubble material, the amount of blackleg in that field, some will blow around in the atmosphere, but most of it will be distributed locally from these guys. So if you get rid of these guys, you reduce the disease pressure. You reduce the amount of blackleg in that particular field. So it is quite possible on a 1 in 4 rotation to grow something like Westar, which has absolutely no blackleg resistance. You wouldn't want to, but as you know, it is possible to grow respectable crops of that variety. But we don't do that, do we? We have, especially my neck of the woods, you know, every other year canola. We have, you know, the landscape is pretty yellow a lot of times, and that's really driven by economics. The trouble with that is it drives that adaptation process. So what do? Well, if we're not going to have a longer canola rotation, we can do things like reduce the volunteers growing in that field. And why is that? Just for fun, or you know, if you know, pretend you're a fungus now, you know, you're floating around in this canola field, which is now the wheat field. Oh, wait a minute, there's a canola plant growing there. I'll infect that little guy. From the pathogen's point of view, you've not gone canola, wheat, canola. You've gone canola, canola, canola. And in other words, you're perpetuating that cycle of resistance. Another thing that could be done, at least in theory, and is yeah.
There's another thing about volunteers. Remember with hybrids, the volunteers that are growing.
Yeah. Okay.
Actually, the resistance is usually only in one parent, and when a volunteer from a hybrid growing, it's actually some of both parents. So 25% of the volunteers usually are susceptible completely back.
Yeah, So did everyone hear that? So what Murray is saying is when you have so when you plant a hybrid canola, which almost all our varieties are, and it produces seed, you have those genes segregating. And then depending on the gene, if it's a major gene resistance, only a quarter of those plants are going to be resistant, and the rest are going to be semi-resistant or completely susceptible. So you're also changing the genetics. You're weakening that whatever resistance that you have. So that's a double no-no, I guess, right? So thanks, Murray. All right. So the other thing we can do other than increasing that rotation, at least in theory, is to change the genes that we throw at the fungus in the field, right? So if a blackleg population has become adapted to a particular set of resistance genes in a variety or a group of varieties, the last thing you want to do is put that same variety or varieties with the same resistance genes back into that field. Why? Because you've done the same thing as not rotating that canola crop or throwing those same genes into it. It's the same as with clubroot. You know, if we only have one type of resistance and we keep throwing those resistance genes back into there, you're really helping the fungal population. So what the Australians have what we think is a really neat system, and Murray alluded to it in the clubroot context as well, of resistance groups. And that's the idea. If I can identify a group of cultivars, a group of varieties that have the same or pretty similar resistance genetics, and I'll call those group A, and I also identified another group of varieties that have different resistance genetics, call it group B, and it's come time for me to book my seed for the next canola crop. What I'm going to want to do is pick a group B, not a group A. And so this has been working in Australia, and one project that's going on right now is to see if we can adapt the system to the Canadian context. Because the problem is right now, I can tell you, well, you know, you should rotate your canola varieties, right? Well, that's nice, Ralph, but to what? I don't know, just something different. All I can say is don't put the same one in that same particular field. So this is going on now. We've been doing some testing in the greenhouses, really just to develop the method of how we're actually going to measure this. But what we seem to be finding is we have something like between 2 and about 5 different resistance groups. And the reason for the wiggle room there is depending on how you calculate it. If you're really stringent, you get a couple of groups, and if you're not so stringent or you use a different method, you get about 5 different groups. If we only have 2 resistance groups, and I can tell you right now, one of those resistance groups is Westar all by itself, so that's kind of useless information, really, right? We have, in effect, we have one resistance group, ain't going to work in the Canadian context, and if that proves to be the case, what it speaks to is the lack of diversity, at least as far as blackleg resistance goes, in the collection of Canadian Brassica napus, Argentine canola cultivars. If we have about 5 groups, that's actually a usable number to work with. Do you remember how many Australians they have? Something that goes to G or something like F or G, and there's a little bit of overlap. So that's under test right now. That's one approach. Remains to be seen in the next couple of years if that's going to work. If it does work, it'll simply be a system of when a variety is registered, it gets tested, it gets placed into a group, and in a publication or in a label or something like that, there'll be group A, B, C, or 1, 2, 3, and producers will be able to manage their genetics that way. It'll be a tool. The other approach is to measure or to characterize what the genetics are of the fungus populations that are in this particular variety or in this particular area, and match that with the genetics of the canola variety. So there's get a little bit technical now, but there's a core idea in plant pathology of the gene for gene hypothesis, which says if a canola variety or any crop variety has a particular gene that gives resistance to a disease, that pest population, that pathogen population has a matching gene that is kind of like a lock and key that'll fit that. And so if I change one side of that equation or modify that, I either do or I don't get disease. So, and if we can sort of cross-tabulate which genetics are present in the host and which are present in the pathogen populations, that'll be a management tool for either the breeding companies or decision makers, let's call them, to target genetic resources in a particular area. So that's kind of a long-term process. That involves a lot of isolation, genetic analysis, and so on. We kind of got a start on that. A lot of work done at the U of M and Ag Canada on characterizing the canola variety side, or getting a start on that. And now projects are running on characterizing the host side. So just as, I guess as a foretaste, I would say that the pathogen populations, judging from some of the populations of the blackleg fungus, Leptosphaeria maculans, in Canada is actually pretty diverse. Why? Because we've got a lot of these guys floating around in the fields, shuffling their genes around. And on the other side, there seems to be some indication, as I mentioned before, that the canola resistances in the inventory of Canadian varieties to blackleg is not that diverse. So what do you do with that? Well, right now, all I can say is manage those volunteers and try to move those genetic resources around. So change varieties. Now, there are fungicides registered for use against blackleg. And so sometimes I get the question, well, I can go on a short rotation, or I can grow the same variety in my field again, right? Because I'm going to put fungicide X on it, and that'll take care of that. Well, one of the people that works in my lab actually did that, even though he knows better, and found out that it's not actually 100% protection. Fungicides will reduce that pressure, so they'll drop the amount of blackleg in a crop down, depending on variables like weather and whether you get it on time and on time and so on. But it is not a substitute for resistance management, crop rotation, and that kind of thing. So it's not a silver bullet.
Hey Ralph.
Yeah.
If a farmer, say in this area, had a 1 in 3 or 1 in 4 canola rotation, would you recommend or would you expect to see results from a fungicide application for blackleg?
So the question was, if someone's using you said a 1 in 3 or?
3 or 1 in 4.
Yeah. So like a respectable rotation, would I see a positive effect, presumably, of putting a fungicide on?
Like a payoff.
Yeah. So the studies, I'll summarize what we know now by saying that if you have a whole lot of disease pressure in a field, so that field is going to get smoked because it has a long history of canola, there's a lot of disease pressure, and you put a fungicide on that, you're probably not going to see an effect. Why? Because it's just being overpowered. And by the same token, if you've been following, so I've been poking around in here, for example, I can't find any blackleg. Is a fungicide going to help here? Well, probably not, because is it so low. Now in Autumn's example, she's saying there's a 1 in 3, like a respectable disease rotation. Is there blackleg in that field? If it was in a kind of a moderate level, yeah, it's gonna push that I would say that's the ideal situation where we would see that population, the blackleg population, go down in response. Sorry, Murray, I sense you want to add on to this topic.
So what about the non-disease effects of a fungicide application?
So Murray's question was, well, what about the non-disease effect? I'll broaden that a little bit. So if you put certain fungicides on to a crop in the absence of any disease, and you can replicate this in a laboratory situation, you'll get a growth regulator effect. And this will sometimes be marketed or discussed. I have seen this myself, and this is my opinion now. I don't think it's that significant. what I think is significant is that if you spray a fungicide on a field like this, so there's no blackleg behind me or very little, but there's alternaria. Later on there'll be gray stem, there'll be a little bit of sclerotinia, there'll be fusarium, oxysporum, any number of things. I call them the nibblers. You will drop the population of those things, and if each of those takes out, I don't know, 0.5% of yield or 1%, well, 1% plus 2% plus 0.5%, pretty soon it adds up to a fair bit, and also your crop will look greener. So you do see positive effects from that. Whether the economics in that situation are justified, comme ci, comme ça, right? I don't think it's proven, but if there's some data to support that, well, data's king, right? I'll believe that. But fungicides are a tool. It's just something that we can add into that that we have in that toolbox. Maybe to close off this discussion, I would say the days of just booking seed, looking for the R label, the blackleg resistance label, and then throwing that out into the field and not worrying about that anymore I think they're over. For blackleg, for clubroot as well. I think the days of easy disease management are shifting into a new era where we're going to have to look at managing. So I am going to just go into the symptoms here, but any questions on the genetic management on the gene stewardship front?
Just sort of backing up to your comment about controlling volunteers. Sure. and related weeds in that mustard family. We've been able to control stinkweed for 50 some odd years now, and we've still got it. Good agronomic practices tell us that we try to control the majority of the weeds, not 100%, because it's just too expensive, and hopefully crop competition will look after the rest. But so, I guess my point is that some of those weeds, some of those volunteers are still going to be around in the field, only just due to good agronomic practices.
Right. So
Current agronomic practices, let me put it that way.
So if I can summarize, you're saying we're never going to get rid of all, in the same way that we've never gotten rid of all the stinkweed, we're never going to get rid of all the volunteers, and therefore we're never going to get rid of all the blackleg population in a particular field. Is that it? So we're always going to be living with it. I violently agree with you on that. But what we're talking about is it's a numbers game. So in the same way, we can't tolerate having a canola or any field full of stinkweed, 50% of the plants being a stinkweed. We can't tolerate having 80% of all the plants infected with blackleg. In fact, with disease at least, we probably don't want all of that fungus population going down. So if I could wave a magic wand and for this year make every bit of blackleg fungus disappear from this municipality, let's say.
You'd be rich.
Well, I'd be rich for the year, but what would happen is well, we would get rid of that, but suddenly we leave the door open for a different population to come in and take over, and suddenly we have a different scenario again. So it's, if you think back to your ecology days in university, or maybe you learned about the lynx and the hares thing. You get a lot of hares, and the lynx population goes up, and then that drops down. It's kind of analogous. You have a host pathogen predator prey kind of relationship, and we are now in the era, as just saying, of sort of keeping those things down to a dull roar, and living with it at levels that we can. And I think that's kind of an ideal situation. So yeah, the magic bullet era of throwing out a resistance variety and not worrying about it. I mean, it was nice, right? You don't. That's one less thing to worry about. You don't have to deal with it. But unfortunately, the world is getting more complicated. Okay, oh, sorry, Ken.
Yeah, I wanted to continue that discussion a little bit. I had the opportunity of touring a number of Australian visitors, and I found it quite intriguing. You know, they were appalled by the fact that we were comfortable with a few weeds, kind of like the comment that Dennis was making, is that our agronomics are based on economics, and that we're not going for 100% eradication. But in Australia, with the amount of resistance development on the herbicide end, they're really targeting complete eradication, and they felt like he was kind of telling me, you know what, you're going to be doing the same thing in 10 years. So on the weed side of things, like they've gone back to hand weeding and
Yeah.
whatever it takes, like a higher level of management, obviously, but they're still focused on the costs. And I think that same type of transition happens both with the diseases, with weeds, and probably with insects and the beneficials that Hector are going to talk about. So I don't know, like, is there, you know, do we wait till it's too late, or do we start maybe thinking about that a little bit sooner?
So the question is, I don't know if everyone heard, are we going to have to go to more an eradication strategy to prevent any opportunity for resistance buildup?
Well,
As in
they
a,
tend to be really awful things to deal with, like, you know, glyphosate tolerant weeds or club root or something like that.
yeah. You may be right, but a thing to keep in mind is that the Australian ecosystem, or the way they grow crops, and it depends on where in Australia and where in Canada, right? But in a general sense, the way canola is grown in Australia is different from the way canola is grown in Canada. So we have a very short season. You know, here you're a bit longer, but in Vegreville, people will be seeding in May, and harvest is in August, September. In Australia, depending again where you are, those plants can sit in that field for 100 days plus, you know, like most of the year. And that plant might be sitting there not growing because it's in the dry season, but it's been infected with a blackleg fungus. That thing is adapting and cycling the whole time. So they've had resistance breakdown, I think in a single season or 2 seasons. And then when they've pulled, depending on the variety and the genetics, pulled it out, they'd be able to go back after a few years and put that in. So their race change, I'm speculating, is going to be a lot quicker than in Canada. That said, we don't fully know the answer to that in Canada either. So I would say on a whole, Australia has been very proactive in understanding their systems, you know, and sort of looking at the basic ecology, the basic genetics of that. And I think we're only now sort of waking up to the fact that we need some of this basic information. So that's also being gathered now, but it would have been nice if that had been done 10 years ago maybe, but I think the Weasley scientist answer to your question is I think we need to understand the ecology of the blackleg system, or I guess in weed systems as well. I won't stray into that too far, and manage accordingly. So it's probably true in Australia they may have to eradicate their weed populations, or really look at suppressing the blackleg fungus in particular areas. We may be able to live with it. All depends. Any other questions on that? I'm just going to wrap up here with a little quiz. Oh, sorry.
This is a sort of quick question.
Yep.
With your bag of your woody stems, you talk about if you can get them to decompose faster. So are you saying maybe tillage is coming back?
No. And I know this from personal experience. So the question is, can we till to get rid of the reservoir of the, and you just know from several studies, including some of our own, that tillage doesn't really help. It helps in some other disease systems, but this one, so you think you run a disc over a piece of ground, it kind of pushes it down. You may run a cultivator over before seeding, or when you're seeding, pull some of this stuff up. You can actually get things sporulating faster if you put them into a slight anoxia, so slight low oxygen levels, and you pull it back up again, and you get more. So some of them may decompose a little bit faster, but on net, you don't actually get a more rapid degeneration. The only thing that really gets it is time, and probably also in areas where you get more rainfall or something, just the environment generally decomposes it quicker. And I think probably also if you have a more diverse ecosystem in the sense that you have a reduced tillage system, where you not only have canola and the blackleg fungus, but you have canola and a whole bunch of crop residues, a whole bunch of other fungi and bacteria on it. You know, there's things that not only eat the canola, but eat the thing that eats the canola. So that's probably a factor as well. And while we're on the topic, burning definitely does not help. Canola fields don't burn very well, but if they do, you end up with a stub, you know, sticking up out of the ground about that tall. And that's where all the blackleg fungus is, and there's no issue there. So really, the panacea is rotation, unfortunately. If I had a magic wand, you know, I'd find something to bust those things apart or to make them more susceptible to degradation that worked reliably, and that might be a way to increase that degradation rate. Okay, who feels confident identifying blackleg in a field? Who can tell it from sclerotinia, for example? No. Oh, one, okay. Yeah, so I think probably blackleg is underestimated because a lot of times people don't know what they're looking at. So maybe come up here after. Do we have a short break after this, or what's the? Okay, I guess, or stop me if you can. We're looking for lesions on the stem or on leaves that have a lightish interior. They tend to have a dark border in the outside, not always, and they tend to have often a chlorotic. That means a yellowed area around the outside. If it's on the stem, especially if it's on the bottom, you'll see narrowing. If you pull the plant, it'll tend to break at that spot, and it's dry. Sclerotinia, if you take a stem infected by sclerotinia, right, it's kind of whitish colored often, and if you push on it or tug on it or twist it, it's soft. Blackleg is a dry rot. Inside those lesions, you'll see a bunch of little teeny spots, which will ooze a pinkish mass of spores under humid conditions. And then this is the important one. If you take a set of clippers like this, and you think you have a lesion on a stem, especially low down, and you clip just below that, and maybe take a few slices, and you see black discoloration on the inside of the stem. Inside of a canola stem is kind of a whitish cream color. If you see black, not brown, but black, that will almost always tend to be the blackleg fungus. So get some of these. I'll hide the brand name. The brand name doesn't matter. But a good sharp pair of these clippers and maybe a loop or a magnifier to take a look at that. Resistance breakdown in a, well, who's missing hair like me or is gray and remembers blackleg in the field in, I guess, the early 90s, the 1980s? Who's seen it? Who's seen a bad blackleg infection before? Not many? Oh, there's Rob. Yeah, Rob's seen it. He's older than I am. So you will see, like with sclerotinia, you'll see premature ripening. You will see lodging of the crop. A lot of times it'll look like it's fallen down. People think it's sclerotinia. You'll sometimes see, the plants laid down. They'll stand up new green shoots and try to compensate for that. But then you go in and you don't see sclerotinia, so you don't see white. You don't see a soft rot. You don't see galls. You see those dry rot lesions with those pycnidia and those pieces of stem. A badly infected fields, and we've seen a few of these now over the last 3-4 years. You'll see 80% of the plants sometimes with blackleg on them, and it'll be pretty obvious that there's a problem. It's just distinguishing one problem from the next. So get one of these guys. Scouting it, you can start to look in fields. I would start looking kind of in the when they're kind of in a full rosette stage and when they're starting to bolt, and you can start to see lesions on the leaves. Or in the fall, again, you can look for these stem pieces, and if someone's interested, come up and I'll show you exactly what to look for. If you see a lot of these stem pieces with the pseudothecia on them, so the sexual spore stage, you know that field has a bit of an issue. And I think I'm getting the hook now. So
Stop you if I can.
okay. So I'll be packing up here. I kind of got a skedaddle, but Ken can put you in touch with me if you have any questions, and Murray knows.
Maybe mention that you're looking for fields too.
Well, so we're as part of these studies, both of the resistance grouping work and characterizing what the fungus populations are, we're looking for fields to monitor, especially in the south, and especially kind of in the Lethbridge, Tabor, Brooks, even Calgary area. And what we'll be doing is going into those fields over the next 4 years and sampling, counting how much blackleg is there, isolating the fungus, and actually sending that to Winnipeg for genetic analysis. Murray?
Does blackleg affect mustards, yellow mustards?
So the question is, will they affect yellow mustards?
Cultivated mustards.
Yeah, so cultivated mustards. So the blackleg fungus, Leptosphaera maculans, will affect a number of weeds and probably most cultivated brassica species, including vegetables, Brassica oleracea, and so on. There is some specialization, although I wouldn't put it past some crosstalk between the two. I don't know the horticultural impact. I myself have, maybe you have, Murray. I've never really seen a wipeout in a mustard crop from blackleg, although you can find it in those. Yeah, well, and so Ken was saying it's in drier areas. If you blackleg goes up in areas where you get adequate moisture. If you start to get flooding, and it goes down in dry years, it kind of parallels sclerotinia in that way.
I think the Gensias, like the Oriental and brown mustards, have a pretty good level of blackleg resistance.
Yeah, but there's variability within the Gensias, and I don't know enough about the mustard varieties, but the canola quality ones are pretty good. But again, it's confounded with the areas that they're grown. The canola quality ones have performed fairly well in our nurseries in Vegreville. So I think if you're a mustard grower, it's not going to be your primary concern, honestly. So before I actually get the hook, thanks for your attention, and we will move on. I don't sense any more questions. So thank you very much. Give us a call if you're interested in talking to us about fields. And we kind of need to line everything up by about harvest time. So swathing stage, let's call it, slightly before or slightly afterwards. And then we need permission to go in and sample your fields, even if they're not in canola, for the next 4 years. And you can contact Ken about that as well, if you like. Thank you.
Well, join me in thanking Ralph.